All right, everyone. Good morning. It's 9 o'clock. St. Mary's webinar series continues today. This is the third in our series. Just remind you, coming up in February, on February 4th, we have a presentation on cover crops, which we're putting together right now, but it looks to be a very strong presentation in that regard. Alyssa's sort of leading that up, and she's been in contact with a number of people, including Shalimar Armstrong, our own faculty member who works on cover crops, and a number of other individuals from the area who will be participating in the cover crop discussion. So that's coming up February 4th, 9 a.m. and 7 p.m., just like this arrangement. So that'll be our next call. Today is about soil health, and with us on the phone, we have a number of our team members. Emily Usher works on our human dimensions aspects of the project. Alyssa Besser, who's works in my lab, and she's has a strong coordinating role in all these videos in terms of getting things kind of going the same direction at the same time. Joe Rorick's joining us today. Joe is going to give us a presentation here in a second or two on soil health. Joe is a major player in soil health in the state of Indiana. He has an extension role in soil health, as well as being a contributing member to a number of the other programs around the state in terms of soil health. Linda Propicki is here. Linda is the recently named department head of horticulture, but she's also better known for her role in working with human dimensions and trying to understand attitudes and things that happen related to agriculture in the state. Ruben Goforth is here. Ruben's an aquatic ecologist and guitar player or collector. I can't tell which. I've never heard him play. I've only seen him on the wall. So I know both things exist in the world, players and collectors. So Ruben is an aquatic ecologist who's been doing work in streams and looking at how different practices and things affect the fish population and microinvertebrate population. Ben Wickard is here. Ben is with the Indiana Ag Nutrient Alliance. He's a big collaborator on these projects. He's been a real asset to us in terms of getting in terms of collaborations and cooperations with folks around the state. So it's been great there. Again, I didn't introduce myself. I'm Ron Turco. I'm on this project as well. I'm also the department head of agronomy here at Purdue, and happy to be here. This is actually my area, one of my areas of expertise is in soil and soil health. So I'm really happy to be having this presented today. I think it's a really exciting area. It's one I think it can make a major impact on ag productivity and as well as water quality, sort of at the back end of this. Also with us today is Sandy Vogelweed. Forgot to see you there, Sandy. Forgot to introduce you. Sandy is a main player in all these, and she's been really helpful in turning in things in the watershed and getting things to work for us up there. So she's been a great collaborator with us. So with that, I think we'll get rolling here. I've taken the approach. We're going to have a video that we have 3 videos today. We have a video that Joe Rorick put together with us, looking at soil health. We have a secondary video that second video that Alyssa Besser put together. And we have a third one that's very specific on some by one of our students, Laura, who looks at soil enzymes, something people have asked us about a lot. So we put together a separate little video on that, just so you can have some direct knowledge of when we start talking about some of these things. What does it look like? How do you do it? Those kind of questions. So we're trying to bring you a bit into the lab in some of these questions. How do we measure these things? What do we do? How do they look? When people talk about these things, what does it mean? Those kind of things. That's really one of our main goals with some of these videos that we put together. So with that, I'm going to, well, I'm sorry, stop 1 second here. We'll show the video, then Joe's going to be with us so we can answer questions specifically. So if you have questions, hold them till the end of the first video, and then we'll have Joe will jump on and we'll let him answer direct questions from the presentation, and then we'll proceed with the next 2 presentations. So if that works for everybody, I will, in my sort of semi-Zoom knowledgeable way, I will get this video going, and so you guys can see. Are there any questions right now? All right, so let me do this. Okay, hit that button, and then I will hit this button. I think I'm sharing my screen.
We don't have audio.
Do you want me to mime along with it? Hi, I'm Joe Rourke, and I'm the agronomist for the Conservation Cropping Systems Initiative. I'm with Purdue Extension. And then you can skip ahead the first minute or two.
Are we having trouble with the audio?
Yes.
Okay, I don't understand why. I never understand why it doesn't work, because it works when I do it otherwise. Give me 1 second here.
We had it the first few seconds.
Really?
If you
Great.
did something, I don't know if you did anything.
I didn't touch anything. Well, I think I did. I think I know what I did. Okay, let me try it again. Let me know if you hear it. Okay.
The field. On the left-hand side there
we go.
we've got a field that has had multiple tillage passes in their system. And on the right-hand side, we have a field that has no-till and cover crops in their system, right? So look at the difference between the clarity of the water coming off those two fields. So that was during a rainstorm. What about when it doesn't rain? These pictures here are from the drought of 2012. Of course, that was a very dry year. And on the left, you can see that soil is bare, that soil is dry and cracked, and that corn's starting to roll. On the right, where that soil has been covered, where that soil has not been tilled and left bare, that corn looks better, the corn is taller, and you can't actually even see the soil, right? It's had some cover there to help to protect it from evaporation and to hold some of the moisture that we did get during that season. A couple more pictures here. These are from central Indiana, and you know, these are pictures. These just happen to be from central Indiana, but this is everywhere, right? And with some of these pictures I show, sometimes people are like, well, you know, it's flat here. We don't have any hills like you showed there. We don't need a water control structure because water doesn't concentrate. It's, you know, it's flat here. Well, here's some flat fields, and even without that, even without the rolling topography there, water still collects, right? Water will still be unable to infiltrate into the soil and will collect on the surface of that soil. So on the left-hand side with heavy tillage, you can see that water ponding there right across the road, even in just a relatively moderate rain, right? 1.8 inches here. There's less water standing there, and even if it's maybe possibly close to the amount that's on the left, when that water does move, right, because that water is not going to stay in that field, that water is going to move, it's not going to carry that topsoil with it because that soil has been protected. It's got those residues there to be able to help hold it in place, right? Last example, and this is an extreme one, but kind of less of an extreme one than we would really hope, right? This is after an 8-inch rain, and it just sort of seems like maybe we're getting bigger rains than we used to. We're getting more extreme rain events than we used to. So here's a set of pictures that same side by side like we've been looking at, just near each other. And on the left, you can see that gully formed, right? So that field is not functioning properly. It's not able to hold, it's not able to withstand an extreme weather event. On the right, where we've got that cover crop, where we've got some good soil cover, it was able to protect that field. There, you know, there may have been a little bit of erosion under there that you just can't see, but we can't know that, and it's obvious no gully has formed where it would have washed those plants out, right? So can you change your management, or can you work with your growers to change their management such that it can withstand some of these heavy rains that we get? So we've looked at a lot about what soil health looks like, but what is soil health? When we talk about soil health, what are we talking about? So soil health is the continued capacity of a soil to function as a vital living ecosystem that sustains plants, animals, and humans. And I've got function And I've got living ecosystem underlined there because they are very important. We often forget that our soils are supposed to be alive, right? And it's the biological properties of the soil that we are only sort of more recently really understanding. We've got the physical and the chemical properties. Those have been studied pretty well for a lot of years. And, we think we have a pretty good handle on those, and we didn't really understand the interaction between the biological part of that and the importance of the biological part on its own as well, right? But it's also the continued capacity of a soil to function, because you don't just get to soil health and then that's it. You have to keep working at these. Now let's take a quick couple of minutes to just look at a way you can go out and check for function in your fields, in your soils, or work with your growers to look for some of these soil functions that we're talking about. These are just sink strainers or bathtub strainers. You can get these cheap, and you want to take soil from that top inch or two of the field or the area you want to look at, and ideally you want to have a comparison, right? So pick an area that's different, either better or worse, you think might be better or worse, to compare it to, just so you have an idea of what you're looking at. On the left, we've got a common cropping system, one crop, maybe two crops with a lot of tillage, multiple passes per year. On the right, we have a soil health system that has a lot more diversity, right? So 2 cash crops, maybe even 3 cash crops, corn, soybean, wheat, for example, or cereal, rye, or oats for grain. And low disturbance. So we're talking no-till here. We're talking strip till, min-till, sometimes maybe. So we're gonna lift these strainers up, and we're gonna sit them in that water, and we're gonna let that soil soak up. And we're gonna talk a little bit about what happens. So right away, as that soil on the left goes into the water, you can see it start falling apart, and you can see it start clouding the water up. That water clouding up is an indication that that soil is breaking down into its individual sand soaked clay particles, and the sand will fall out of suspension pretty fast, but the silts and the clays take longer. And if that water leaves, then it washes those silts and clays with it, right? And you can even see a little bit of particles floating in there. That's going to be your organic materials. So your silts and your clays wash away, and your organic materials that are floating in there are also going to wash too. On the right, you can see that soil holding together. You can see some particles falling, but it's not clouding the water up. That's indicating that the aggregates that are in that soil are able to hold together, right? That soil is biologically glued together to be able to withstand the water as it wets up, right? So once you let that soak up for 30 seconds or a minute, you want to lift them up. You want to let any free water drain, and you just want to tilt it out onto a surface, right? So I've got it out on the table here, but I've used the bed of a pickup, shovel works. And you want to take a look at what happens. On the left, that soil is not able to hold together, as indicated by the water clouding up and the particles falling, right? Here's a zoomed in view, a little different angle, but you can see that soil breaking down, and you can see that water starting to make its way out. That's a soil that has a bad relationship with water. That's a soil that's not functioning properly. On the right, you can see that sort of granular structure still. You can see those aggregates in that soil there. And if we change views a little bit, this was the same test. You can see the same setup there, but I ran it twice to get a little different angles. So that picture on the left there is the screens again with the soils in it. The picture on the top right is the same right after I dump it out. And you can see the differences between how that soil either muds up and clumps together, or is able to hold itself and stay that nice and crumbly granular structure. And then this picture on the bottom right here with the permanent marker for scale there, on the left, you can see that soil This is after it's dry. So this
It's dried
dried,
just overnight, so maybe 12 hours or so. One of these soils is ready to take another rain, and one of these soils is not. This soil on the left here of that bottom right picture is crusted. That one is not going to be able to take another rainfall until that dries out enough to crack open, and then maybe it'll be able to infiltrate some rain. But that soil on the right dries just as loose and open as what it came out of that screen as. So this is this slump test I would encourage you all to try, because that's a really good, fast, easy, cheap test to go out and look for some of this soil function. We already mentioned that soils have biological, chemical, and physical properties, and that they interact in a complex way. So how do we then overlay soil health onto those complex interactions? So with the physical properties, really what we spend a lot of time talking about and what we're really concerned about is the physical disturbance. Of course, there can be chemical and biological disturbances as well. A really, really heavy manure application or something or sludge can really cause some problems in a system. But really what we're the most concerned about is the physical disturbance, right? So less ground engaging equipment, less soil disturbance. The chemical can be lime, it can be pH, right? You need to make sure that you're have those right. Salinity can also play a factor sometimes in some areas. We don't really have that here specifically, but and then the biological. We already talked about how important those biological properties are. But you want to be able to add or grow more organic matter. You want to be able to store carbon in your soils. What are some indicators of healthy soil? You want that soil to absorb and infiltrate water. We talked about that. We saw the water ponding on those pictures. You want a soil to be able to retain water, and but that's not to say you want soil to retain water the way that we often think, right? That most of our soils retain water and they just turn into swamps when we get too much rain. You also want adequate aeration, right? So together with retaining water, you want adequate aeration. You don't want that soil to crust up. You want that soil to be good and friable and have good aggregation. And if it doesn't have good aggregation, it may have a crusting issue, and it may not retain water properly, and it may not have good aeration. Often you can dig up plants, or better yet, you can dig up cover crops, roots, and take a look, see if they're doing what you want them to be doing. You want to look for, J roots, thickened roots, things like that. You just want to have an idea of what's going on in your soil. We have soil health principles that we use to guide our decision making, right? Because soil health is all about management, and it can be complicated, but so we have these 4 principles to use to help guide our decisions when we're thinking about soil health management. You want to minimize disturbance as much as you can. You want to maximize soil cover, you want to maximize biodiversity, and you want to maximize the living roots that are in that soil throughout as much of the year as possible, right? And it's important to understand that the more of these you can do, the faster you'll go on your soil health journey. But maybe in your operation or in your situation, you're not able to do all these all the time, right? So if you know that something's going on in a field or on a farm, and maybe some tillage will need to be done, and you can't necessarily minimize disturbance, but then maybe you need to maximize soil cover in some other way to help account for that sort of thing. But let's go through these in a little bit more detail. Minimizing disturbance, again, we often think about this as physical, and that's what I'm going to spend my time on. They can be chemical, they can be biological in some cases, but really, the bulk of our disturbances are physical disturbances through regular multiple pass full width tillage. Here's a picture that shows some different implements that are used, right? So you've got a low disturbance drill, you've got a high disturbance drill. Clearly some differences there in how much disturbance is done, right? Because we're looking at the relative bare soil that you can see. You got a disc harrow, and on the far right, you've got a moldboard plow. So it's just important to understand when you're trying to minimize physical disturbance that the implements that you run through a field and the way, the speed, the depth, how they're set can all affect the disturbance. And even if it is done as minimally as possible, if it's not done properly, it still can be a problem. What we're looking at here, these two pictures are from a site that has had a vertical tillage tool run through it. They've run a VT tool twice a year, fall and spring. And even though it's a shallow pass and it's not a very aggressive pass, particularly, especially compared to a disc or something, if you look at that radish root on the left, down at about 2-3 inches, right where at the bottom of that tillage pass is, that root had a hard time penetrating that plow pan that has been formed, right? So when that VT tool has been run, conditions either weren't right for that, or it was set too aggressively, or something, or a combination of factors, something went wrong, and we're starting to get some compaction here, and we're starting to get a restricting layer that is going to be a problem if conditions are right, or if conditions aren't right. So on the right hand picture, you can see that root growing horizontally and all the little hair roots coming off of it also growing horizontally, so that layer is going to be restricting, especially if it turns dry, or conditions aren't just perfect for growing a crop. So do we have a restricting layer here? Yeah, sure, we do. And you need to look out for those. And if you're going to do tillage, or if you're not going to do tillage, then you need to understand the differences that's going to happen in your systems. So this slide shows a graph on microbial activity. And this blue spike here is, after tillage is done and the soil warms up, those soil microbes start really going crazy and chewing up all those broken up aggregates and all that free carbon that's been accessed because those aggregates have been broken up by tillage. And then you get this crash once they use all that. And often we think about in our systems, that's when we'd be planting, and we take advantage of those nutrients to the best that we can, but we do end up losing a lot. When those seedlings are small, they're not taking up a lot. So the free nutrients are lost from that system. When you don't till the soil, it takes a little longer to warm up. Of course, you don't break open all those aggregates and open up all that soil to let those soil microbes take advantage of the stored nutrients and the stored carbon. So you get this sort of plateau that throughout the season you get a slower, more consistent release of nutrients. But if you're counting on that spike, this is a big change. So, this is why we talk about in reduced tillage systems, we talk about how important, starter fertilizer is, for example. You want to maximize soil cover as much throughout the year as possible, both with crop residues and ideally with cover crop residues as well. That cover is protection. That cover is protection from raindrop impact. That cover is protection for wet harvest conditions, right? Because if you leave that soil intact, if you don't break it up, that soil has more structure for you to drive on. And then, like in this example here, it's got living roots growing in there to hold that soil, to help hold that soil together. So even if it turns wet here, that soil is going to be able to hold up a combine better than if it had been a tilled system, right? It's going to help protect from temperature swings. And often we think about this in the spring, but we need to start thinking about this more into the summer when those soils are really too warm to be functioning properly, as well as in the spring, right? It's going to help protect from evaporation. So if things turn out dry, and we often get dry summers, or at least periods of dry spells in the summer, the more water your soil can hold appropriate for crop for that crop to use, right, the better off you'll be. And it's also protection for the soil life. And remember that soil needs to be alive. That soil's supposed to be alive. So we've got crop residues that provide a passive blanket, and we've got cover crops that can provide sort of an active blanket, right? And these two pictures show what happens when you don't have enough cover. So these are both from the same field. Actually, these are some plots with and without cereal rye as a cover crop. And these are all good no-till. But on the left, you can see that soybean residue was not able to protect and hold that soil in place. We had a little disturbance with the planter, which of course you have to do, right? You need to plant a crop. But then they got a hard rain, and that soil washed out of those seed slots, and it washed out some of the seeds out of those slots. On the right, where we had a little more cover there, it was able to protect that soil, and it didn't have those same problems. You need to actively manage for water stable aggregates, and this is a big part of soil cover. So these two soils are also from it's a different field, but these two soils are from the same field. These are sandy loam soils even. And that same slump test that we saw earlier, both of these plots had a cover crop over winter, but one was tilled in the spring and one was left with that cover on. And then we came back in the fall, and you can see on the left where it was tilled, that soil had crusted up. Even a sandy soil had crusted up. On the right, that soil still had good aggregation. It still was nice and friable. And you can see they acted differently in the water. Not to just stick to corn and soybean systems, soil cover, it can have big implications on how much soil splashes too, right? If that soil isn't able to hold together in water, when a raindrop impacts that, it will splash and it'll move that soil. And if you've got a fruit on the ground there, it's going to get all over that fruit, which can help to spread disease. It can cause a lot of problem. That pumpkin's going to need to be cleaned off before it's going to be as marketable. But on the right-hand side, where that pumpkin was grown on a mat of cereal rye, you can see it's nice and clean. That one can get cut and put on the truck. Maximizing biodiversity is our third principle that we want to look at, and you need to remember that you have to feed that soil herd, right? So an acre of soil, you'll see a lot of different numbers for this. This is just happens to be one of them, and I like the graphic. But an acre of soil, healthy soil, has the equivalent of 2 cows worth of microorganisms living into it. That's an underestimate. That's an old figure. But I like the picture. But you just need to remember to feed that soil herd, and you. So what we're looking at here is a bunch of different cover crop roots. This graphic is available on our website, but you can see all these different kinds of cover crops. These are just some common ones we have that we're able to find good representation. You can see all the different kinds of rooting structures our common cover crops have here. And we zoom to a real example, there on the left-hand side of the picture, you can see that root, and it's covered in soil. Right, that root has been exuding sugars. It's been exuding carbon, and the bacteria and the soil microorganisms have responded to that. And there's a little ecosystem forming right around that root. Those are hot spots for biological activity. That root you can see in the middle is that's a hairy vetch root, and you can see some little bitty nodules forming on it, right? So that's going to be fixing some nitrogen. You can see an earthworm in there, right? So as you feed that soil, as those cover crops feed that soil, then you start to create ecosystems in there, and it starts to cycle and it starts to function more. And with that, we just talked a little bit about that, right? Providing continuous living roots. Those roots feed the soil. We just saw that, and here's that a little bit differently, right? You can see that those corn plants fixing sugar, fixing carbon from the air, making it into sugar and pumping it into the soil and using it for itself, right? We've already talked about how important a living root is to feed the soil. But in addition to those roots actively feeding the soil, when they're alive, they will also feed the soil and create pores after they die, right? So here is that same a part of that same graphic we saw, and zoomed in a little bit on fewer species of cover crops, so you can see some of the different rooting structures there. But as those roots die, they will then leave the channels that they're in open, which then gives an opportunity for, the crop that's planted after those to use those same root channels that are more nutrient dense because they've had the roots growing in them. But oftentimes, a lot of earthworms, a lot of soil microbes will be living in those, and that's where our nutrient cycling is going to be the hottest, is in these rooting areas, in that rhizosphere around those roots, right? So we have our 4 soil health principles to help guide our management decisions. And again, you just want to do as many of these as possible, as often as possible. So you want to minimize disturbance, you want to maximize soil cover, you want to maximize biodiversity, and you want to maximize living roots as often throughout the year as possible. To conclude this talk, I'd want to run you guys through one last example. Here we have an aerial image of a cornfield from our Infield Advantage program. And we all know light green corn is bad and dark green corn is good. And what we're looking at here is that dark strip in that field on the left-hand side of that picture, right there where the arrow is. What could that be? Is it, is it soil type related? Is it something went wrong with the fertilizer rig? What happened there? Um, if you look at the overlay of that soil map, though, on the right hand side, you can see that used to be an old field edge. So we get that dark green strip of corn there where there used to be a fence row, right? And if you think about it, that fence row has had our soil health principles inadvertently applied to it, right? It's had minimal disturbance. It has had soil cover maximized by all the plants growing in it. It's had diverse plants growing in it, so our biodiversity was maximized, and it's had a living root growing in it throughout the year. So our living roots were maximized, right? So we applied those principles to that fence row, we didn't specifically do it, but they were applied there. But what happens if we can apply our management principles beyond that fence row, right, across the whole field, to get that dark green strip of corn to be the whole field and instead of just one little strip, right? So that's what we're talking about when we talk soil health, right? We want to apply our management principles to our fields to build our soils and build their function, right? We need to bring our function back to our soils. So my name is Joe Rorick, and here's my contact information. Please feel free to reach out, and thank you guys for your attention.
All right, Joe is actually with us. You guys maybe heard a couple of times in there. So Joe, if you have questions, let's get some questions going.
I have one question to start. How do I do the big voice?
What?
How do I do the big voice that came in there a couple of times? That was great. I want to do that more often.
Yeah, I'll figure that out for you. I'll let you know. A little bouncy on the video, I think. Questions?
Hey, Joe, I've got a question if nobody else does. I really liked your example of that vertical tillage. You showed those images of a compacted layer kind of in a vertical till. In our project, we, at least on the soil side, we kind of have a range of farmers who are using various tillage practices. And we have put people kind of in categories of kind of high, medium, and low tillage. And vertical tillage was one that we had real kind of difficulty with in where that is kind of on the scale. And of course, our putting people into practice groups kind of, it takes into account 4 years of tillage practice and cover crops and things like that. But where would you put vertical till? Like that is a low impact tillage, correct? And so where would you put that kind of on a scale of impact, I guess, of tillage, soil health impact?
I mean, it is low impact as things go, right? If you're holding it up against a disc chisel run 3 times or a moldboard plow, it's a lot less intensive than that. The issue is more in what is called a VT tool, and how they're set, right? So the angles of the discs can run anywhere from 0 to 5 or 7 degrees. Some are even a little more, 10 or so, depending on the manufacturer of the equipment. And in that example, it really just comes down to, sometimes sometimes you have to do tillage. There's some good reasons why tillage might be done. But if you're gonna do tillage, it's really gonna be important that you do it when conditions are fit for it, and that you're not gonna cause more issues than what you start with, right? You want to identify why you would want to do that tillage, and then you want to go out and do that tillage so that it helps to resolve the issue that you're trying to resolve by doing that tillage. But yeah, so a VT tool, I mean, it really can be minimum disturbance, but you can set them to where they do a pretty fair bit of disturbance.
Okay, thanks.
Other questions?
There's some questions in the chat. Joe, can you see that? Do you want to go ahead and answer those? Looks like Don's posted a couple comments, or maybe they're just comments and you want to comment again.
Yeah, absolutely. Thank you, Sarah. So Don, I think I got a little at your root exudates comment in the slides just following when you made that comment. I did see it come through. But yeah, the idea is you put a plant out there. If it's your crop, great. If you don't have a crop in the field, you put a cover crop out there, and it pulls carbon out of the air. It fixes carbon from the air and uses the sunlight that's available all year. I mean, most of the year. Some days it doesn't feel like we get very much sunlight, but it's available all year long, and pumps that carbon into the plant and then also into the ground and feeds that soil. And yeah, taking a look at the soil after you run a VT pass, if you put your hand in there, or if you dig down, move that soil away, and start to look, see if you have any of these hard layers. In longer talks, I go into just going out and digging a hole, right, and having a look in your soil and seeing if you have some of these limiting layers or these compaction layers, because they can be a big problem that we don't necessarily think about always. And tillage may not be the answer to those either, right? But if tillage is going to be the answer, just do it as carefully as you can.
All right, any other questions right now? Okay, well, let me put the next video up. This is part of our project, the St. Mary's project. Alyssa's been working on putting together some of our information from the project. And this video is really about some of our findings, really. This project's supported by IDEM, and it's also supported by US EPA. And we have many other contributors, a long list of people from Indiana who are contributing, organizations that are contributing to this project in terms of interest in protecting water quality in the area, as well as the water quality in Lake Erie. So that's really where some of the justification for the projects come from. Let me show you this little video, talk specifically about the St. Mary's region and some of our work up there. And I'll get it going right now.
Share the screen.
Hi, my name is Alyssa, and I'm a graduate student at Purdue on the St. Mary's project. Our soils team looks at a variety of soil health indicators, but they all fall into 3 categories: chemical, physical, and biological. I'll talk about those today. I'll give an overview of the whole soils project. We'll talk about the data we've collected thus far and what it means for soil health. Our team takes samples from approximately 25 sites in the Nicholson and Weber Ditch subwatersheds, which are both a part of the Western Lake Erie Basin. Each site consists of 3 replicates. Each season, we sample from a different place in the field and use GPS coordinates to check the soil type against the soil survey. When soils are collected from the field, we first measure 3 10 by 10 meter plots, each 15 meters apart. Once the replicates are in place, the team will take 6 soil cores from each replicate. This core is 4 inches in diameter, and we sample to a 10 centimeter depth. We'll take 2 soil moisture tins, and finally, a bulk density from each replicate. And here we see Becca taking a bulk density sample, and this can be a really difficult process.
Okay.
Here, she's just finished hammering that piece into the ground, and she'll remove the sleeve, and she's going to cut between those 2 cylinders. The sample is that bottom cylinder, and if she cuts too much off, or if she's hit a rock or a root or the sample crumbles, she'll have to start again. Bulk density tells us a lot about the soil, its compaction, and the soil pore space that allows for movement of water, nutrients, and microbial habitat. When finished, tools are sterilized to ensure we do not cross-contaminate the soil samples between sites or take pests from one site to another. Samples are then taken into the lab where they are processed and stored. As I mentioned earlier, our lab measures a number of different soil health indicators. Biological indicators would include microbial respiration and enzyme activity, whereas chemical indicators would be things like pH, nutrients, and organic matter. Lastly, physical indicators would include textures and aggregate stability. In addition to these soil health measures, we've also collected farm practice data. If you're a participant in the St. Mary's Project and we take soil samples from your field, you've probably met Marianne or myself as we've surveyed farmers about cover crop and tillage data for the last 4 years. Using this information, we've been able to put participants into one of 3 categories. P1, or Practice Group 1, are participants with minimal use of conservation practice. That's a conventional tillage, and they do not grow any cover crops. P3 are farmers who use no-till and cover crops, as well as a nutrient management plan. P2 is a middle group of participants, maybe moving into conservation, or those who implement some conservation practices and not others. Using this practice data, we've been able to compare the data of the 3 groups. So what story does the data tell? The data collected thus far indicates a strong correlation between farmers of high conservation practice and microbial activity in their soil. This is substrate-induced response, and it measures the microbial reaction to glucose, a food source. The higher the microbial activity, the higher the nutrient turnover may be in the field, which leads to a biologically healthy soil. This is important to soil health because an increased active microbial population not only increases nutrient turnover, but also contributes to other soil health factors like soil aggregation, seen here, which also increased in the high conservation groups. This is aggregate stability as mean weight diameter, which is just the size of the aggregate. A strong aggregate stability would decrease erosion and nutrient loss, aid in better soil structure, and again, promote that microbial activity. Lastly, phosphorus sorption increased with high conservation practice. This is a soil's ability to retain phosphorus, and it means limiting movement of phosphorus off-site. This is a particularly interesting find and one we're looking into further. Thanks for watching. If you have any questions about soil health or the St. Mary's Project, I can be reached at abesser@purdue.edu.
All right, so Alyssa's with us, and can answer any direct questions about this, but it actually really does support nicely some of what Joe was talking about, what we're seeing in the watershed in terms of activities and work that's going on. Is that we are seeing very big impacts from conservation on processes that go on that are important. So it's conservation is paying off. Joe, have you seen this in other places?
Yeah. So imagine that, our soil health, all this soil health that we talk about is real. So we find this pretty consistently in a lot of areas, especially with aggregate stability. And that's why we talk about that slump test that I showed, which is that aggregate stability test that Alyssa talked about, which is a fairly time-intensive lab test as far as that goes, but that slump test takes 2 minutes, and you can run out and check that really fast. And actually, we do have soil health diagnostic kits in every county in this state. It's usually your soil and water office or your NRCS office, but if failing that, it may be at your extension office. And any of this stuff that you would like to try to do, especially the infield stuff, let us know and we'll come out and do it with you.
I think it's important to remember that the microbes in soil, and there's, like, as we've seen already today, the microorganisms in the soil are tremendously high number, right? So they're very much there. There's lots of them in soil, millions and millions of them. They're not very active normally if you don't have a carbon source. What's the carbon source that we see in soil? We see plant exudate, root exudates. And that's really where the cover crop comes in. That's where the active biomass comes in. When you start adding those plants back in, you excite the microbes. And you excite the microbes, you basically cause them to start building up the organic matter. The organic matter leads into soil structure. So this all ties to the physical aspects. And in my opinion, as a soil microbiologist, that's what I am. The microbes really are what drives soil, right? So I'm claiming credit now for all the good things in the soil are driven by the microbes, but it's really the physical properties are impacted by the biological activity in soil, and if you can increase the biological activity in soil, you can improve your soil physical structure, and that's really a key piece of what Joe was talking about, what we're seeing related to conservation. And everything else that goes on in soil is really about this active biological property in soil that's driven by inputs of carbon via the plant source. Microbes, mostly, not all, some can do this, but don't directly interact with CO2 and bring down CO2 on their own. They have to go through a plant step where there is a capture that goes on via photosynthesis. And that's where that tie together comes from with the microbes in the plant. So. Other questions?
Hey, Ron and Alyssa, I've always been excited about the P3 and the phosphorus story. And this leads a lot into Ron's comment about how microbes are the ultimate, rulers of the soil health domain. So I am, it is an interesting interplay with phosphorus because phosphorus is governed by microbial functions, but it's also really affected by physical factors and chemical factors. So it's a really tough question. I know we don't know all the answers just yet, but I'm curious for the group, your comments, and maybe Joe and Alyssa and Ron can kind of talk about more like, where's the next steps in understanding really what's driving phosphorus, particularly under these cover cropping kind of scenarios.
Well, I'll let Joe answer that one.
Thanks.
You're welcome, Joe.
Yeah, and it is a great question. And we have historically looked at phosphorus in terms of the chemical and physical processes. And I'm not a soil microbiologist, right? I'm a field agronomist, and I do not understand all the biological interactions with phosphorus, right? So we think they're chemical and physical interactions, but what happens when we, in the cases of our really good high-end soil health systems, when we totally upend the physical and the chemical processes with our biological ones, what does happen with that, right? Does that change our phosphorus cycling? Does that change our potassium cycling? Does that change what's available in the soil? and when. We don't really know a good portion of that. And that's why at this point, what we do know is if you have a living root in that soil taking up available nutrients, it can capture those and hold them in the field, right? So that's where we're at now. And that's why we talk so much about cover crops. We think about it a lot with nitrogen too, right? Because nitrogen's really mobile, but it's happening some with some of our other nutrients as well, especially, you know, sulfur is also mobile and things like that. But you know, just try to keep what you have in your fields, both by keeping the soil in the field and by keeping what's in the soil tied up in a
You know,
plant.
the other thing that
cycle.
happens, Joe, and you show it nicely in some of your images, is that, and it goes to what Sarah's talking about, is that as you improve soil structure, you improve the number of small holes, you improve the organic matter distribution in the program. You put everything gets spread around a little bit. That's very retentive for phosphorus. You can hold phosphorus that way. You're going to have a certain core mineral component in soil that really is the base of what goes on with phosphorus, right? There's certain physical attributes of that soil that you're not going to change. Your iron aluminum ratios, et cetera, which have a real big driver in phosphorus retention, but you can dramatically alter that with the biology, right? You can add more organic matter, you can add the roots, like you're saying, it acts as a biological trap. Microbes take up phosphorus. Fungi take up phosphorus. They hold it. They're like a little plant in the soil. They need phosphorus to grow. It's part of ATP, it's part of proteins, it's part of their cell walls and membranes. So a bigger active population will retain phosphorus in those systems. So that's, I think, a really important dynamic here in terms of looking at the impacts, and especially at a regional level in terms of water quality and phosphorus, is that the biology is active in that system and helping to hold on to the phosphorus.
Yeah,
We have
I
one
like
more
to think-
video. Sorry.
Can I say one more comment thing?
Yep.
I think that was a totally stage question clearly, but I like to think of it as like almost like a synergy. Like we're creating almost a greater impact than each of those systems could do on their own, because by the microbes being there, they're doing all those things of uptake, but they're also creating the soil structure that's more spongy, more sticky for phosphorus as well. So they're acting to enhance some of those physical components that already exist and taking up on their own. So it's just like the sum is greater than the individual components kind of
Correct.
situation.
We have one more video. We've been asked a lot about this question, and Laura, one of our undergrads, got real excited about this area and worked on this she's in the background there with Alyssa. Worked on this as a project, as an undergrad thesis kind of little project, and we're going to show the video. It's on soil enzymes, right? So a lot of people are interested in soil enzymes, and remember, soil enzymes, and she'll explain this better than I do, really the expression of microbial activity. Microbes to eat is the key piece. When they eat, they create enzymes to dissolve, pre-digest, if you will, food from soil. So they create the enzymes, and we measure the enzymes as part of the microbial activity assessment. So if they're active, they have to make enzymes to be active. So that's really why we look at enzymes in the soil. So let me get this video.
Dr. Turco, as you do that, we had one last question in the chat. Don, you asked about infiltration. And infiltration was something that we were taking early on in the project, in the first couple of years in the project. And as time went on, that just became a really time-consuming effort. We only took it once a year and only in kind of certain fields. And at the end of this project here, we are on year 4. That was a piece that we just dropped because there wasn't enough data points to kind of say anything about it, and it's an all-day kind of measurement. And so that was something that you asked about infiltration, and that's it.
Thank you, Alyssa. Let me show this video, and Laura's around. Here we go.
Hi, my name is Laura France, and I am a student at Purdue University studying agronomy. I'm working on the St. Mary's Soil Health Project. Our soil team collects samples from agronomic fields surrounding the Weber and Nicholson watersheds of the St. Mary's River in northern Adams County in northeastern Indiana. This is part of the Western Lake Erie Water Basin. In this video, I will be discussing enzyme activity in soil, how they affect soil health, and how they are affected by farmer practices. To start off, enzymes are everywhere. They are involved in mammal digestion and metabolism, plant photosynthesis, decomposition of organic matter, and so much more. Enzymes are catalysts that speed up the rate of biochemical reactions. Without them, reactions that would normally take less than a second to occur would now take decades to happen or not happen at all. Our soil lab analyzes 4 different enzymes. They are acid phosphatase, alkaline phosphatase, beta-glucosidase, and beta-N-acetylglucosamidase. In this presentation, I will be referring to them as AP, ALKP, BG, and NAG. AP and ALKP are both a type A phosphate enzyme, but they operate at different pHs. AP operates at lower pH while ALKP operates at higher pH. This trend can be seen in these graphs. AP activity decreases with increasing pH, while it is the opposite for ALKP. Phosphates are involved in many things, but in general, they are enzymes that remove phosphate from a protein. Different proteins will result in different consequences of removing the phosphate. In plants, phosphatase is involved in respiratory carbon metabolism and the hydrolysis of organic phosphorus into inorganic phosphorus. This makes it available for plants to uptake this essential nutrient. The enzyme BG can be used as an indicator of microbial activity in the soil. The main job of BG is to hydrolyze cellulose. Hydrolysis is the chemical breakdown of a compound due to the reaction with water. Cellulose is the main substance in the walls of plant cells, which helps plants to remain stiff and upright. The hydrolysis of cellulose done by BG results in the sugar molecule glucose. Glucose is a carbon source for microorganisms. In simpler terms, BG helps make the food for microorganisms. NAG has a complex role in cells. First, chains of complex sugars called oligosaccharides are formed in the cytosol of cells. Cytosol is the aqueous component of cellular cytoplasm where various cellular particles are suspended. NAG releases particles called N-glycans, which breaks the bonds of oligosaccharides, allowing them to break down into simple sugars. This process is called catabolism. This overall process results in cells being able to retain the maximum amount of simple sugars, which are then used as energy within cells. These enzymes play very important roles within soil and plants. By comparing the total enzyme activity of each enzyme to different farmer practices, we can see which practices result in higher enzymatic activity. Our team has categorized our sample fields into 3 different groups based on the amount of conventional and conservation practices used. P1 group includes all conventionally tilled fields. P3 group includes fields that use little to no tillage and plant cover crops. P2 includes fields that implement a mixture of these 2 practices, such as tillage with cover crops. Our data shows that fields that had some form of conservation practices had higher enzymatic activity compared to those with no conservational practices. On a different scale of categorizing conservation practices implemented by farmers showed that a mixture of conventional and conservational practices yielded the highest enzymatic activity within soils. The scale used goes from 0 to 2. The more conservation practices implemented, the higher number that was assigned to it. For example, the field that tilled in spring and fall and did not plant cover crops would be a 0. A field that was no-till and planted cover crops was assigned 2. Our data shows a correlation between farmer practices and enzymatic activity within their agronomic fields. This is because farmer practices directly affect soil health. For example, heavily tilled fields may create a fine seedbed for crops, but they break the natural structure of soil and create soil compaction after heavy rainfall. Soils that have been compacted lack the space for water and oxygen, both of which are necessary for microbial life. Soil microbes and other organisms such as earthworms help to create healthy soil. The healthier the soil, the better the soil is able to retain nutrients. Another example is the use of cover crops. Cover crops also add organic matter into the soil and reduce the risk of erosion from water and wind. Not only was enzyme activity affected by conservation practices, but also by the crop planted. Grain, including oats and wheat, correlated with soils having higher enzymatic activity compared to fields that had a previous crop of corn, soybean, or preventative plant. This is because plants play a very important role in soil health. Plants contribute to the amount of organic matter within soil. Plant roots produces substances containing substrates, which are molecules that react and bind with enzymes. Based on our data, it can be inferred that grain crops produce more enzyme-rich substances than other crops. Including a grain crop in crop rotation could increase enzymatic activity within soils. Thank you for watching this video. I hope you enjoyed it and learned something new.
All right. Any questions on soil enzymes? We have the experts with us.
I have one that's hopefully quick, but may not be a pleasant one. Was that prevented plant treatment a planned one, or was that a fun little factor of how the year went?
One little factor of how the year went.
The preventative plan had not been planned. It was just over that summer when fields were flooded and it just got too late to plant any crops, so farmers switched to preventative plan.
Yeah, we got that dropped on us, Joe, you know.
Yeah. Oh yeah.
Field work, you gotta love it. Other questions? Let me check the chat. Sandy's got some stuff about Adams County in there. In terms of getting some help on nutrient and soil testing, great. And then Don got his question answered on infiltration. Yeah, and then we talked about the fungi. So yeah, I think we're pretty good. If there are no other questions.
Ron, if you go up a little bit further, Marty has a question about cover crops. She posted it at 9:52.
Let me see, hold on. 9:52, Marty. At least in the future, using cover crops and non-native, it can become invasive. It moves the soil they particularly like. Joe, you want to?
Sure. Yeah. I mean, it can always be a risk, right? But that's why we talk about having, especially with cover crops and with soil health systems, you have to really intensively manage these, right? I'm sure you guys have heard the old joke about, grain farmers having 4x4 trucks because they farm 4 weeks in the spring and they farm 4 weeks in the fall and they spend 44 weeks a year in Florida, right? We all know that's not true anyway, right? But you have to intensively manage these, and so the idea is you need to be able to get those cover crops under control, and you want to have them under control. Of course things happen, but we don't. I mean, cover crops have been around a long time, and we do not see them moving into natural systems. And we don't find them in natural systems. And of course, annual ryegrass can be one that's difficult to control, and it is one that we worry about, but with proper management, it's not one that we tend to have an issue with, right? So that's just goes along with all of these. We just have to increase our how intensive we manage to make sure things keep in control.
Great segue to our next set of presentations on February 4th, which will be on cover crops. And we're bringing specific cover crop experts into the discussion. And that we'll lead off with that question. That's a good question, Marty. And then we'll have them talk a little bit about that in terms of how cover crops play in these environments. So yes, that's our plan for next month, and we'll jump right into cover crops then. Other questions? All right. I appreciate you all coming out for this. This is great. I think we've had a great turnout this morning. We're going to redo this tonight at 7. If you want to learn even more, you can come back and hear the same thing over. It's repetitive, but we get a very good number of people involved that way, and we'll be back at 7 tonight, show some videos and have some discussion as well. So if you're interested in, come on back. Otherwise, we'll see you next month for a very detailed discussion on cover crops. Thanks for coming.
Thank you.